The universe is a womb for life, which is suspended in infinite nothingness. This is our reality, as seen by Dr. David Nichols, a master pharmacologist, organic medicinal psychedelic chemist, creator of molecules, student of consciousness, and all-around master of the craft of chemistry. I would personally say this guy is kind of a hero of mine, and it is an absolute treat to be able to get this masterclass from him, which I consider a masterclass of consciousness, psychedelic chemistry, pharmacology, the mind and matter connection, and the potential healing and medicinal therapeutic application of psychedelic medicines, both artificial and organic, synthetic, man-made, and naturally made, nature-made. So, if you don't know who Dr. David Nichols is, he was at the University of Peru and was the only person who had a DEA permit to be able to synthesize controlled substances in his laboratory, and that enabled him, he just happened to be kind of in the right place at the right time and have the right mindset to take what was obscure and occulted information, the dark ages of psychedelics. Basically, this man took psychedelics out of the dark ages and put them into the culturally sanctified, establishment-driven limelight. Like, he took psychedelics out of chaos and ignorance and heresy, and he put them not on a pedestal, but he gave them their rightful place in the universal pharmacopoeia, I'll call it, like, of things that simply are medicine. Whether they help people or not, there's a lot of debate over what we're calling medicine, how we're defining it. This man took psychedelics and placed them into their proper position as therapeutic tools and as medicine for the individual, which has laid the groundwork for this psychedelic renaissance that we're seeing now. It's probably goes without saying that, if you're not familiar with Dr. Nichols, he wrote the introduction to the book P-Cal, A Chemical Love Story by Alexander and Anne Shulgin, which they're known to communicate back and forth quite a bit. And they were studying at the same time. However, Shulgin was underground. David Nichols was, I guess you could say, above the ground or on the ground. And he was mapping out the psychedelic experience from a biochemical standpoint, from the standpoint of biology and chemistry. Now, this type of knowledge is going to be immensely powerful in the days and the years to come, because what they're finding now in these intravenous DMT sorry, that's a big topic. I'm just going to try to introduce this man, right? He synthesized Schedule 1 substances before anybody else had ever done it. And it'll probably never happen again because the restrictions and the regulations are getting way tighter. But this is before any regulations. And he had a Schedule 1 substance. At Purdue, he synthesized DMT for Rick Strassman. He synthesized psilocybin for John Hopkins. He synthesized like 40 kilos of MDMA for some research he was doing at the school and employed his students and stuff. Like, this man has legitimized a type of medicine which is far more powerful than anything you've probably ever seen in your entire life. There's not a lot of things that change your life. There's having a child, getting married, moving somewhere completely new. Still not that much of a change, but psychedelics You know, when somebody's born or when somebody dies, that changes your life. When you take a psychedelic, it is that magnitude of of change, that magnitude of an event which alters your life permanently. And so it is an admirable feat. And if you look at this man's history, he comes from a working class, worked his way to the top, excelled in all his courses, and he absolutely earned everything that he claims. And he doesn't even claim that much. He just is a very humble human being, I think. And he is quite a master of the craft. So that's, I think, everything I have to say to introduce him. Like, let's see what else. He wrote the introduction to PCAL, and he founded the Hefter Institute, which was an institution designed to study traditional uses of these medicines and put them into the clinical literature. And this is research that is paramount to the evolution of our species. This master class, I think, represents a milestone in human evolution that will be looked back upon as sort of like discovering the holy grail of the consciousness question. And that being said, it's not the philosopher's stone. There are still many pieces to put together to comprehend exactly everything that is being transmitted through mass reports of psychedelic endeavors. But the capacity for individual healing, for miraculous, mystical, individual healing and direct experience of God has been legitimized by David Nichols, Dr. David Nichols. And I think that in the days and the years to come, that we're going to look back on this time and recognize that we really were in the third great enlightenment, the third great renaissance as it is happening. And we just don't conceive of it now. But if you're not convinced by what I'm telling you now, then just watch this masterclass on pharmacology, psychedelic chemistry, the mind and the matter connection, and a basically user's manual of the world's most popular, powerful, and talked about psychedelic medicines that we know of and that have been invented. Because, we've got books on books of compounds these people have invented. This chemist, he worked, Dr. David Nichols, he worked with Shulgin. He worked with Anne, God rest her soul. He worked with, what's his name? John Hopkins. Rick Rolfin, I forget his last name. Rick Strassman. Okay, so with that being said, please enjoy this masterclass given to the medicinal plant. With that being said, please enjoy this master class of psychedelic chemistry given to the Medicinal Plant Chemistry Club at Northern Michigan University. I forget which date, 2020 something, 2021, 2022. And shout out to my professors and my teachers, the club leader, and everybody who participated in manifesting this event, because it truly was a treat. I hope you enjoy it. Have a grand existence.
Okay, can people see that? We're good? Okay, so I'm just this is sort of an overview, soup to nuts, starting with some plants that have psychedelics, and then going on to talk a little bit about the pharmacology and how psychedelics work. So psychedelics, as you all know, probably know, are very unusual compounds. This definition was from Jaffe's chapter in Goodman and Gilman's 7th and 8th editions of Pharmacological Basis of Therapeutics. And these are drugs that produce changes in perception, thought, feeling, etc., that are not or cannot be experienced otherwise except in dreams or at times of religious exaltation. That's obviously a very unusual definition for a class of pharmacological agents. Recently, there's been sort of a renaissance, I call it a renaissance in psychedelic research. This is the number of publications on psychedelics, and you can see they peaked around 1970, just around the time the Controlled Substance Act was passed and Nixon's anti-drug war. And you can see publications dropped off to a minimum here in 1995, 2000. And then they've increased. This goes to 2020, but that number has continued to increase. It's exponential. So there's a lot of interest, a lot of people working on it. A number of universities have psychedelic research centers set up now, a number of clinical trials. As of about a month ago, there were 116 clinical trials of psilocybin registered with clinicaltrials.gov, which lists all of the FDA-approved clinical studies. So it's really taken off. There's always been a lot of interest, but there's more interest now, and there's been some funding for this work, and so a lot of people have gotten into it. In terms of where they come from, there are major natural sources of psychedelics. On the left, we have a little peyote cactus. This is probably a year or two old. I grew some of these from seeds years ago. This is Lophophora williamsii. And it has a simple trimethoxyphenethylamine called mescaline in it. So this is a 3,4,5-trimethoxyphenylethylamine. And aerial ethylamine is a very common motif you see in natural products that interact with neurotransmitter receptors in the body. You know that norepinephrine and serotonin and dopamine, they're all aerial ethylamines. They may or may not have a methyl here, and depends on what the aromatic system is. But this is a common motif in drugs that interact with neurotransmitter receptors in the body. These are tryptamines, another class of psychedelic. If you don't have anything attached here, just simply the naked indole ethylamine is a compound called dimethyltryptamine, or DMT. DMT occurs throughout the plant kingdom and also is produced in trace amounts in the human body. If you have a methoxy at the 5 position here, where this R is a methyl, so we have a methoxy here, that's 5-methoxy-DMT. That occurs in a number of natural sources, but particularly recently we've seen it exploited in terms of the Bufo alvarius, the western cane toad, or the Sonoran desert toad. It has that toad has parotid glands on the side of its head that produce 5-methoxy-DMT in combination with a bunch of toxic glycosides. So what people have been doing is catching the toad, and if you squeeze those parotid glands, they'll squeeze out the toxin. So it's a defense mechanism for the toad. If a predator like a coyote would bite the toad, it would spray this toxin into its mouth and probably would kill the predator. But if you smoke it, they'll spray the toxin out on a glass plate or mirror, and then when it dries, scrape it off and then smoke that. And in the process of smoking, it breaks down those toxic compounds, and only the 5-methoxy-DMT is volatile. And it's extremely potent psychedelic. And then we have the so-called lysergamides. They're semi-synthetic, and they're derived from the ergot fungus, claviceps. This is Claviceps purpurea infecting a stalk of rye grain. And these little black things are called kernels of ergot. They're actually the sclerotia, or the inactive or dormant form of the fungus. And if you would put these into a culture dish where they would have the nutrients to sprout, they'll actually grow and put out little fruiting bodies, little tiny mushrooms. And they produce, by and large, compounds called ergolines, or in particular lysergamides. An ergoline is a 4-membered ring system these four rings here with this indole as the central nucleus. Again, you see an aromatic system, 2 carbons away from a basic nitrogen, and that's a common motif. Lysergamides are amides of lysergic acid. So lysergic acid would have a carboxylic acid here at the 8 position, and LSD would be the diethyl amide. So these R groups would be ethyl groups. The stereochemistry here, the more active one is the 5R, 8R designation. And so these would be active lysergamides or lysergic acid amides. Ergot was responsible for ergotism throughout the Middle Ages. And during the Middle Ages, more than 40,000 people died from an epidemic of ergotism. So in the fall, if the weather was cool and damp, it would be conducive to the growth of ergot fungus on the grain. And grain was a major staple of diet in the Middle Ages. They didn't often have meat and vegetables, so bread was a major staple. So if they made bread from grain that had been infected with ergot, the ergot alkaloids would be absorbed, and then of course you ate bread every day, and so you had a chronic exposure to ergot alkaloids. And they produced gangrene of the extremities. So ergot alkaloids, some of the ergot alkaloids are potent vasoconstrictors. They cause the constriction of peripheral blood vessels in the fingers and toes, produce nervous spasms, burning sensations, hallucinations, and was called temporary insanity. People just their cognitive processing was really screwed up. And ergotism was also called St. Anthony's fire. St. Anthony was a patron saint of people that suffered from ergotism. They had an intense burning in their extremities. And these are examples of gangrene in the toes and in the fingers. So this is kind of what would happen. And people that survived their bout with ergotism often would be missing fingers and toes. If they survived, the fingers and toes would become necrotic and drop off. So you could see those people when they'd survive. And ergotamine is one of the common ergot alkaloids that would be produced by the ergot fungus. Again, we have the ergoline ring system. These 4 rings down here fused together, a relatively planar molecule. And depending on the ergot alkaloid, this is ergotamine, but you had ergotoxin and a number of other ergot alkaloids, which differed only in the nature of this substituent attached to the amide nitrogen. In Mesoamerica, the Aztecs, they used mushrooms. I actually didn't talk about the mushrooms when I showed that. I talked about the methoxy at the 5 position. If you have a phosphoryloxy at the 4 position, that would be psilocybin, and that occurred in the so-called magic mushrooms. So in Mesoamerica, when the Aztecs couldn't find the mushrooms or they were out of season, they often would use the seeds of Turbina crombosa, which they refer to as ololuki. This is a species of morning glory, and an ergot fungus grows symbiotically with the seeds of ololuki. And so it produces ergot alkaloids in the seeds. So you could grind up the seeds, macerate the seeds, mix them with wine or some other solvent, and drink that and get an effect. And that doesn't have LSD in it, but it's got lysergamide, just the primary amide here. I've circled it. So you don't have any substituents here. This is not very potent. People in the 60s, when they couldn't get LSD, would go to seed stores and buy an ounce or 2 of morning glory seeds and grind them up and eat them. And I hear it's a fairly unpleasant experience. I never did it. But the ergot alkaloids are in the seeds. Hawaiian baby wood rose wasn't used by the Aztecs, but it's in the morning glory family. It also has a symbiotic relationship in the seeds with the ergot fungus and produces ergine or lysergamide. The one you may have heard about a lot is ayahuasca or huasca. Huasca contains DMT as its principal psychedelic component. So if you look at this plant, Psychotria viridis, which is called chacruna by the Mesoamericans, it contains DMT in the leaves. If you try to eat the leaves or you make a preparation of DMT, it's not orally active. It's broken down by an enzyme in the liver called monoamine oxidase. And so it's inactive when administered orally. However, in the Amazon Valley, you also have this vine, Banisteriopsis caapi. And if you cut the vine down, you break it down and shred it, This is the red wood you see here at the bottom. These are shredded pieces of Banisteriopsis capivae. On top of Psychotria viridis leaves, you boil these two together for a prolonged period of time, and you get this brown extract. You filter out the plant material, and that extract is ayahuasca. So what happens is the beta-carbolines in Banisteriopsis are reversible inhibitors of monoamine oxidase in the liver. So essentially, ayahuasca is an orally active form of DMT, and it lasts about 4 to 6 hours. So you may hear of people going to Peru, participating in ayahuasca tourism, and some people talk about taking ayahuasca. There are even places in the US that have these sort of underground therapy centers where they will administer ayahuasca. Ayahuasca is also used by some churches. The União de Vegetal, which is a Brazilian church, it started Brazil. There's a branch of the church in the United States, and also the Santo Daime, which is also a religion, and they use ayahuasca as their sacrament. There was a big court case I testified in years ago where the church, the União de Vegetal, they had a maestri, or a sort of the equivalent of the priest, living in New Mexico. He established a branch of the church. He went to Brazil and learned how to speak Portuguese and learned the church dogma and started this church. And they were shipping it to the United States, and the Drug Enforcement Administration knew about it, let it go for a while, but eventually they stopped it and said, you're importing a Schedule I substance, we're not going to allow you to do that. So they had a big court case because this was the sacrament for their religion. So basically the DEA was taking away a necessary component of their worship. And the court ruled, all the way up to the Supreme Court, ruled in favor of the church under the Religious Freedom Restoration Act. So if you're a member of the União de Vegetal or the Santo Daime, you're legally allowed to go to church services and use this. But if you're a student at Eastern Michigan University, you'll get in trouble with the authorities if you try to use ayahuasca, or if they catch you with it. Just to give you an idea of doses and durations, mescaline is probably the least potent of the psychedelics, the one that comes out of the peyote cactus. 200 to 400 milligrams of the sulfate given orally lasts fairly long, 8 to 10 hours, but it's very prototypical. It has it's noted for its beautiful colored visions, synesthesia. You listen to music and close your eyes and see colored patterns. So, but not very potent. LSD is probably the most potent of the psychedelics, with between 0.1 and 0.3 milligrams of the tartrate salt given orally. It also lasts about 8 to 10 hours. Psilocybin, which is magic mushrooms, it's about 10 to 30 milligrams orally active, lasts 4 to 6 hours. And of course, people eat the mushrooms themselves, the dried mushrooms, or they chop them up and put them in some mixture and eat those. So 3 to 5 grams of the dried mushrooms will give you an effective dose of psilocybin. The DMT is the tryptamine without any substituent on the aromatic ring, 30 to 100 milligrams, generally smoked. Again, it's not orally active. 5-methoxy-DMT is extremely potent. Sometimes it's been called the God molecule, or sometimes it's called toad. 10 to 15 milligrams, either nasally insufflated or smoked. Very short action, 15 to 20 minutes, much like DMT itself. And there are a whole variety of phenethylamines made in my lab and by Alexander Shulgin, which are usually orally active, 2 to 50 milligram doses, and have variable durations of action, usually 6, 10, 12 hours. Now, the interesting thing about psychedelics is their effect depends upon the set and setting. Your mental expectation is the set, what you think is going to happen, and the environment where you take it. So, for example, if you had read a lot of literature about how psychedelics could produce religious experiences, transcendental states, and so forth, and you read a lot of that literature, and that was the way you'd been thinking, and then you took a dose of one of these and went to your church or synagogue or whatever and took it, there's a high probability that you would have a religious experience or transcendent experience, something that would be mystical and very powerful. And when I used to tell the pharmacy students, when I talk about set and setting, I'd say, if you take the same dose of the same drug and go to a Friday the 13th or Freddy Krueger movie, you probably will not have a religious experience. So most drugs don't depend upon set and setting, but psychedelics really depend completely for their effect upon what your expectations are, how you prepared, and the environment where you took it. Just to show you how LSD was first made, or essentially how it could be made now, It was first synthesized in 1938 by Albert Hofmann working at the Sandoz laboratories in Basel, Switzerland. And what you can start with is an alkaloid like ergotamine. And this is an amide. And if you know any chemistry, if you treat amides with sodium or potassium hydroxide and cook the dickens out of them, you can cleave the amide and then reacidify it, you'll get lysergic acid. So this is the way it's done. Hoffman actually treated it with hydrazine, made a hydrazide, and did some other chemistry to get it. But then you can take lysergic acid, and then in the presence of various kinds of amines, diethylamine in this case, with a condensing agent, you convert that into the corresponding amide. So this is lysergic acid diethylamide, and this is an acronym for the German name. Lysergsäure. Säure is the German word for acid, which means sour. That's where we get our name sour, because acids are sour. Lysergsäure diethylamide. He first accidentally ingested it in 1943. So he made it in 1938, sent it down to the pharmacology lab, and they reported back it didn't look very interesting for the assays they were running. So it was abandoned. But he was sitting in his laboratory in April of 1943, 5 years later, and for some reason he just had the idea that the pharmacologist they had missed something that was important about this molecule. He can't tell you what it was. The German words that he used were translated to mean peculiar presentiment. He just had an intuition that the pharmacologist had missed something. So you can imagine if you were a pharmacologist working in a lab and somebody had sent you a compound, you tested it and said, okay, we're not interested in this. And 5 years later, they came and said, I think you screwed up somehow. You missed something that was important. I've got another sample. You can imagine what the pharmacologist would tell him. They'd probably just say, go to hell, Albert, But that isn't what happened. Instead, he started getting this peculiar intoxication, and he left and went home early from work. And he thought, well, this is really weird. He had this 2 or 3 hour intoxication where he saw these beautiful kaleidoscopic colors and images. And he thought, what the dickens was I working with in the lab that caused this to happen? He decided it must have been LSD because he prepared a new sample of this to send down to the lab. So somehow he thought maybe some of this had gotten into his body. Now, he was a very meticulous chemist, and I've talked to him personally. He has no idea how he got LSD in his body, but somehow he did. So he came back on the following Monday on April the 19th, and he took 250 micrograms. Now, I don't know if you know anything about doses of LSD, but 250 micrograms is a pretty stiff dose. And he found out pretty quickly. He was in the lab and he was writing a diary of what happened. And at a certain point, shortly after he took it, he had to leave and go home, and he got his assistant to follow him in the bicycle. It was during the war, and they were rationing gasoline. So he pedaled home on his bicycle. His assistant followed him. He thought he was going really slow, but his assistant said, no, you were pumping pretty fast. They sent for the doctor. The doctor came, checked him. His blood pressure was fine. His heart rate was okay. He seemed okay, but he was just having these intense, colorful visions. So that's when he discovered that LSD was active, quite by accident. And then he went in and he told the research director, natural products director, what had happened. They couldn't believe him. They said, that's impossible. And he'd taken 1/4 milligram because there was no drug known then that had effects at a dose that small. So the people in the company thought, this is not possible. But none of them took 250 micrograms. They all decided to only take 80 micrograms, just in case Albert Hofmann was right. And they discovered that he was right because 80 micrograms is also a pretty good dose. What was he trying to make? So he'd been working with ergot derivatives, and they're very tricky molecules to work with. They oxidize easily, they're air and light sensitive, heat sensitive. And there was a drug called choramine, also called nicotinamide, that was a respiratory stimulant. So people would tend to kill themselves by taking overdoses of sleeping pills, which were barbiturates. And they were killed by paralysis of the respiration. So he thought if we made an analog of choramine and we attached it on top of this ergot nucleus, he knew these ergot alkaloids were quite potent, doses in the 1, 2, 3 milligram range. He thought if he made an analog that had this diethyl nicotinamide on top, that maybe he would get a really potent respiratory stimulant. Of course, that's not what he got, but that was the rationale for his chemistry. It made sense at the time. Now, LSD was an important molecule, not just because of its potency and what it did, but it also was part of the catalyst that spurred a neuroscience revolution. in the 1940s and up to the discovery really of LSD into the early 50s, if you had a child that suffered from schizophrenia or bipolar disorder or some other psychiatric illness, they blamed it on the parents, most often the mother, because the mother was the major caretaker. So there was a term that was known as refrigerator mothers. These were mothers that had been cold and distant and hadn't been nurturing to the children. And that's what they blamed these illnesses on. They really had no concept of biological psychiatry or any idea that brain chemistry had anything to do with behavior. Now that seems strange to us today because we know different, but back then everything was focused on Freudian psychoanalytic theory. So any kind of a mental illness was something that was caused by the parents. So LSD's effects were discovered in 1943, and Sandoz began sending it out shortly after that, saying that it produced a model psychosis. They thought initially what LSD did was produce effects that resembled the effects that what happened in a schizophrenic's brain or mind. And so they were giving it to people. You could just write to Sandoz and say, send me some LSD. And if you were a clinical psychologist, or really if you were almost anybody, if you were a rock musician, you could probably get it. And they would send samples of it out. It was quite different then. And so people had been looking and studying LSD for about 10 years. And there was a woman named Betty Twarog that was working in a laboratory. And she had developed, as for a PhD project, had been looking at a contractile muscle in an edible shellfish called a quahog. And that muscle in the quahog, when you dissected it out, would contract in the presence of serotonin. And so in that era, they knew that serotonin was mostly in the gut. So in the human body, about 85% of the serotonin is actually in the gut in enterochromaffin cells and also in blood platelets. In fact, the first samples of serotonin that were used for structural analysis were derived by concentrating down 10,000 liters of clotted cow blood from a slaughterhouse. So that's where they got the serotonin. So once they established the structure, it was synthesized, and then Betty Twarog was using serotonin as a standard, so she could do a standard curve, put it in with this muscle from the quahog, and then measure the concentration by running a standard curve and then extracting various tissues. So she was taking all kinds of tissues from animals and plants and whatever, and just determining whether or not they had serotonin. So she wanted to analyze brains. She went to the research director and said, I'd like to analyze some cow brains to see if there's any serotonin in the brain. And he poo-pooed her. He said, there's no serotonin in the brain. Everybody knows all the serotonins in blood platelets, and in the enterochromaffin cells in the gut. But go ahead, make yourself happy. She extracted cow brain, and sure enough, it had serotonin. So this is the structure of serotonin. It's a 5-hydroxytryptamine. Again, the indole nucleus, 2 carbons away from basic nitrogen. And you can see how it's embedded in the structure of LSD. Well, you don't have to be a chemist to look at these two structures and say, there's some similarity there, obvious similarity. So the year after, 1953, she published this. In 1954, there was a hypothesis that LSD worked, and everyone knew about LSD by then, that LSD worked by blocking the effect of serotonin in the brain. And then about a year later, they changed that to the hypothesis that LSD worked by stimulating serotonin receptors in the brain. Even though they didn't know what serotonin receptors were, this similarity was the basis for it. 1953, chlorpromazine, I think, was introduced in psychiatry in 1952 or 53. So really the middle 1950s was the beginning of our understanding of biological psychiatry, the neurochemistry of mental illness and normal and mental behavior. And so LSD was a contributor to that, not just the discovery of chlorpromazine, but also the finding that LSD had these powerful effects that resembled to some extent psychosis, and that there was serotonin in the brain. So LSD was important for that reason as well. So how do these psychedelics work? So for phenethylamines like mescaline or tryptamines like DMT and psilocin, they principally activate serotonin 2A and 2C receptors. LSD also activates serotonin 2A and 2C receptors, and the tryptamines also activate serotonin 1A receptors. But the thing that they all have in common, all these different chemotypes, is they activate a type of brain receptor, a G protein coupled receptor known as the serotonin 2A receptor. And that's been established in animal models, in vitro, and also in humans. So if you give humans a drug that blocks serotonin 2A receptors and then give LSD, you can prevent the effects of LSD. So we're pretty sure that that's the major receptor. Now, LSD is a complex molecule that does all kinds of other things. So it's not unlikely or unreasonable to suggest that maybe interaction with some of these other receptors could modulate the overall effect of the drug. But the principal receptor, the essential required but not necessarily complete effect, is reproduced by activation of serotonin 2A receptors. So where is the serotonin 2A receptor? So these were PET studies that were done with PET ligands imaging human brain, living human brain. And this was a ligand that was used to image the serotonin 2A receptor. And what you can see is that the 2A receptor is the most widely expressed serotonin receptor in the brain. It's expressed in the cortex, in the temporal lobes. It's also in the visual cortex. And if we slice through the brain and look at it, this is a sort of an indication of where the expression is highest. High expression is red, low expression is blue. And you can see it's in the outer layers of the brain, in the cortex, throughout the prefrontal cortex, through the parietal cortex, and into the visual cortex. And if you go in and use microscopy and look at this, what you'll see is that the receptors use the antibodies for the receptors. They're expressed on what are called the apical dendrites of cortical pyramidal cells. You can't quite see it in the slide, but these are triangular shaped cells, and they're called pyramidal cells because they're shaped like little pyramids. And the part of the cell going into cell body, this is apical dendrite, and that's where the 5-HT2A receptors are highly expressed. This is the mouse somatosensory cortex. Just to blow that up a little bit, this is the outer layer of the cortex. The dura would be out here in the human brain, and it goes in layers 1 through 6 as you go toward the interior of the brain. And the apical dendrites are in layer 5, and at the bottom, layer 5A to 6, these are the pyramidal cells. The red traces are excitatory branches, and the blue are inhibitory. And these are stacked throughout the cortex, all the way around the brain. And you can imagine that these are like processing units. If you think about a computer architecture where you have parallel processors, your cortex is loaded with these parallel units packed in, where the computational units are these pyramidal cells. So they're, in a sense, they're like the central processing units of cortical processing. And the brain is packed with these, and you can see all the connections they make. So all the information that comes in from lower cortical centers involving things like hearing and vision and taste, etc., they all get fed into these cells, and then they work to somehow integrate that information to produce our sense of what consensus reality is. Now, these receptors look like this. This is an actual a receptor, a G protein coupled receptor. This is the 2A receptor that's been modeled, and they're alpha helices. I've shown the surface rendering of the spheres of the atoms themselves transparent, so you can see the alpha helical backbone. This is actually the receptor, and the neuronal membrane would span the width of that membrane. So this would be the extracellular region of the membrane, where a drug would come in and interact here. And this is all interior, inside the neuron. And these are heterotrimeric GTP binding proteins. So we have a G alpha, a G beta, and a G gamma subunit. So what happens is when a psychedelic comes in and interacts with the 2A receptor here, it changes the way these alpha helices are packed together. It changes their bundling. So they're packed together in the neuronal membrane. It changes the shape of the way they're packed together. And when that happens, it changes the intracellular loops that couple to these GTP binding proteins. principally G alpha A. Normally it binds GDP, and when this happens, when this conformational shift occurs in the receptor, it leads to a change in the conformation of G alpha, and that extrudes a GDP and binds GPP, guanosine triphosphate, and also the G beta and gamma subunits stay together, but they dissociate from G alpha. So the G alpha goes and activates enzymes inside the neuron, and the beta gamma goes and activates other signaling molecules. So the receptor itself is actually kind of like an on and off switch, but it can couple to these bigger units inside, which actually produce the signals. So here again, this is the beta receptor. It's also a G protein coupled receptor, GPCR. The blue, the darker blue, is the inactive receptor. The red is after a beta agonist is bound. And what you can see is helix 5 and 6 shifting out away from the centroid of the receptor. And these intracellular loops are what change their orientation, and the coupling of the G alpha subunit then it's uncoupled from the receptor and does its signaling business. This is just a schematic of that, showing we have the 7 alpha helices inserted into the neuronal membrane. The ligand comes in here, change and activates, it binds in the top of the receptor, changes the bundling or the overall shape of the bundle of the receptor, these 6 of the 7 alpha helices, and that leads to a change in the intracellular loops. And those are what couple to the G alpha subunits or the GTP binding proteins. They also can interact with G protein receptor kinases. And so if you have a phosphorylation here, they'll put a phosphate onto the C-terminal end of these receptors, and that will recruit arrestins. Arrestins are signaling molecules that lead to receptor internalization, also travel to the nucleus, can lead to a variety of signals coming out of the nucleus. Psychedelics like LSD rapidly downregulate these receptors. So if you take LSD on a Monday and then take the same dose every day, by Thursday it won't have any effect. And what happens is LSD, when it binds to this receptor, strongly recruits beta-arrestin, and that leads to a receptor internalization. So the receptor removes itself from the neuronal membrane to the surface, so there's no longer a receptor there for the LSD to interact with. So you get this rapid tolerance after repeated LSD called tachyphylaxis. And that's different from things like opiates that produce tolerance, but you can overcome that tolerance by giving more and more and more of the drug. With psychedelics, you can't. The receptor is simply gone from the neuronal membrane surface and can't therefore interact with another molecule of psychedelic. This is just to show you, this is a tool developed by Kim et al. From our lab and Brian Ross' lab called TruePath. And so these are cells that contain all the G alpha subunits. So I said it's a heterotrimer. They're G alpha, G beta, and G gamma subunits. These have all these different G alpha subunits, all the different ones that are expressed in HEK, human embryonic kidney cells. And so what you can see, these dose response curves that are black are from neurotensin. They transfect these cells with neurotensin. Neurotensin receptors bind to all those different signaling pathways. But if you look at the G alpha Q family, they're activated by psychedelics, serotonin 2A receptors. And so you can see serotonin, LSD, and there's an N-benzyl compound here called an N-BOM. So these are activated by psychedelics in the GQ family of signaling molecules. These are the canonical signals, or the signals everybody recognizes are activated by serotonin 2A receptors. And of course, none of these others are showing any effect other than the standard molecule, the neurotensins, which activate all these different G proteins. So just to show you, you can get different signals from the same receptor. I'm going to talk just briefly about characterizing psychedelics. And there are 2 actual things that you can use to characterize them. One is called a saturation assay. The other is a competition assay. A saturation assay requires you to have a particular drug that you want to look at. So for example, if we wanted to look at receptors for LSD, which would be 5-HT2A receptors, that would require us to have radioactive LSD, and that's available. It's made tritiated. There's also a compound called DOI where they make an iodine that's radioactive. And you can therefore use those by using increasing concentrations in your receptor preparation and then measuring how much binds to the receptor and how much doesn't. And that'll give you a direct assessment of the affinity of your standard drug for the receptor. And once you've done that, then you can then do competition assays, because it's not very easy to get radioactive molecules of every kind you would want. So you wouldn't want to have to make radioactive mescaline and radioactive psilocin and radioactive DOM, and all these molecules radioactive. So once you've done a characterization and a saturation assay, you can then use that ligand to do competition and indirectly determine the affinity by displacing the tritiate LSD. So this is rapid and inexpensive, and it's what all the drug companies use now. Once they have a target identified, they'll do a saturation assay to identify the number of receptors and the affinity of the standard ligand, and then they'll use that and take all the molecules in their library to see how efficiently they displace it. So that would give you an indirect measurement of affinity. So I'm not going to go into this in detail. I've just kind of gone through it. It would look something like this. You'd have your tissue preparation here. In the old days, they would use homogenate, a rat brain homogenate or a pig brain homogenate. We used both of those in my lab years ago. And they had serotonin 2A receptors in there. So you do homogenate, put here. Now you'll use cells that are transfected. So human embryonic kidney cells or Chinese hamster ovary cells that are transfected with the receptor of interest. You put in the radiolabeled drug, you let it incubate. For a direct saturation assay, that's all you'd put in there. But once you've done the characterization, then you would put in different concentrations of test drug, allow them to incubate, come to equilibrium, and then you filter it on a glass fiber filter mat, and then you see how much sticks to the filter. That represents radioactive drug bound to the receptor. So in the saturation assay, increasing concentrations of your radiolabeled drug would lead to increasing concentrations here. And in your displacement assays, you would see as you increase the concentration of your test drug, you would see less and less of it bound. So that would be a measure of how effectively it was displacing your radioactive ligand. And you get a competition assays would give you curves like this, where you would start off with a known amount bound, and then as you add the displacing ligand, you're increasing the concentrations, you displace it. So this is binding, radioactive binding in counts per minute. So that would be displaced as you increase the concentration. And you know that dose response curves, the farther to the left they are, the more potent the drug is. The farther to the right, the less potent it is. So this is just showing several drugs that are displacing the standard drug. This would be the standard drug with a 2 nanomolar Ki, and this one would be a drug that had 50 nanomolar. These are plotted on log scale typically, which linearizes the competition. In addition to measuring affinity, you also are interested in how well the drugs activate the receptor. A lot of times people just think about, oh, it's got high affinity for the receptor, it must be potent. You can have very high affinity drugs that don't activate the receptor. So affinity and efficacy are two different things altogether. So if we look at dose response curves, these are just like typical. A full agonist like serotonin itself, we'd put it in increasing concentrations of serotonin, and whatever we were measuring as our output, when it was saturated, it would reach this height, or it would be a sigmoidal curve, and it would kind of peak out here at a maximum. So this would be essentially the 100% activity for the natural ligand, serotonin in this case. Now, if we put other receptor ligands in there for the same receptor, and they're not as efficacious, they don't reach this 100% maximum, then these would be called partial agonists. LSD is a partial agonist at the serotonin 2A receptor. It only activates it to the extent of about 30 to 40% of what serotonin does. If you have a full agonist and you put in an antagonist, which doesn't do anything but bind to the receptor, then you have a direct competition, and it'll shift the dose response curve to the right in a parallel fashion. So this would be a full agonist plus a competitive antagonist. This would be a silent antagonist, which wouldn't do anything. But of course, if we put in concentrations of a silent antagonist with a full agonist, it would shift it to the right. It's not clear that there are really any silent antagonists anymore. because most antagonists turn out to be what are called inverse agonists. So most of these G protein coupled receptors always generate some signal without any ligand in there. It's like a car idling or something, I guess. And so if you put in inverse agonists, they generally have the structure of what we believe to be antagonists, and they will shut down that baseline activity of the receptor. So we have agonist, partial agonist, what would be silent antagonist, and inverse agonist. So these are functional measures. So if you're producing cyclic AMP in the 5-HT1A receptor, serotonin would do something like this, would produce this level of cyclic AMP. And then if you put an antagonist in, it would mean it would take more of the drug to produce the same effect. Or if you had an inverse agonist, it would shut down baseline production of cyclic AMP. And most of these are done now with robotics. When I was doing this work, we had hand pipetting, and we had it was very difficult. And you can imagine if you're doing a lot of drugs, you really want to minimize your error. And by having robots do this, robots are reproducible. They can be programmed to produce the same amount all the time. So almost all this work now is done through high throughput robotics, using either 96 or 384 well plates. I showed those test tubes earlier. They used to use test tubes, not anymore. Now they use these, which have, sub-milliliter volumes. And then, of course, the radioactivity is read in a beta count or something like that. So now the question is, how can a receptor signal change consciousness? So we know that no animal can model the richness of a human psychedelic experience. If you have a beta receptor and it blocks the heart action, slows down the heart rate, animal models will tell you that. But if you're talking about a drug that changes consciousness, how do you actually do that? And in physiology or pharmacology, we basically use drugs that perturb physiological systems. So if you want to know how a beta receptor agonist or antagonist, what it actually does, you can actually put it into a mammalian system and look at blood pressure, look at heart rate, or look at, put in something that affects insulin production, look at glucose. But what about studying consciousness? Generally, the way pharmacologists study a process, a physiological process, is to use a drug that perturbs consciousness. And that, for example, psychedelics are really great at doing that. The guy who did a lot of this, who's kind of famous, is a guy named Alexander Shulgin, Sasha Shulgin. And he made about almost 200 of these compounds. Some I sent to him, which he included in his books. He published 2 books, one called PCOL, phenethylamines I've known and loved, and TCOL, tryptamines I've known and loved. And he would make these compounds and start at a low dose, take them himself. And if it had an effect at a certain point that he thought was interesting, he would get his wife, Anne, and he would give her a dose of it. And if she thought it was interesting, he had a whole group of friends and colleagues that lived in the Bay Area. And every so often they'd get together at somebody's house and he'd say, okay, I've got drugs weighed out for all you guys. Let's take it and see what happens. And of course, they weren't illegal. It maybe was a little bit crazy to do that, but they weren't illegal drugs. So anyway, and then they would give him a report as to what happened. And then he put those reports into those books, PCOL and TCOL, describing what the average dose was and what the consensus opinion of the group was in terms of what they did. Well, I was at Purdue University, and unfortunately, or maybe fortunately, I don't know, I couldn't, I always had graduate students and people that find out what I was doing and say, do you need anybody to test your drugs for you? And I'd say, no, I can't do that, So we had to use rats. And so research in psychedelics without using humans, we principally use rats, and we use something called 2-lever drug discrimination. So these are chambers, and I've cut away 2 sides, the front side and this inside here. They have 2 manipulanda here. These are little levers that are hooked to microswitches. This is a food delivery pellet system with a house light. And we can reward the rats for performing appropriately by giving them food pellets. So what this system would deliver is a 50 milligram sucrose pellet. Sucrose is just sugar. And so rats like sugar just like humans do. So what would happen is rats don't normally press levers. That's not something that's in their behavioral repertoire. So we'd put him into a box that only has one lever, and then the rat would wander around what he's supposed to do. And if he stood on one of these levers or pressed it, he'd hear a click from the solenoid and it would deliver a food pellet. And he'd stick his nose in here and he'd go, there's a piece of candy in here. So then he would wander around the cage and maybe he would step on it again. He'd hear the click from the solenoid. He'd stick his nose in here. There's another food pellet, another little sugar candy here. So if you do that for a while, the rat learns that if he presses the lever, he hears a click and he gets a food pellet, little rat candy. And then we switch to two levers. Now what we do is we couple one of these levers with the drug. So for example, let's suppose we want to train this rat to press the right lever if we've given him an injection of LSD. And this technique is very sensitive. It produces behavioral effects at doses way below anything that changes the behavior of the rat. So we'll say we'll give him LSD and we'll turn on the right lever and we'll keep the left lever turned off. And now he wanders around the cage, he presses the left lever, nothing happens, but he presses the right lever, he hears that familiar click, there's a food pellet in here, a little rat candy, so he goes and eats the candy. And they wander around the cage quite a bit, they explore, they stand up, they stand on these things, they press them. But eventually, over 2 to 3 months, once you do this, this rat will learn whenever he's given a dose of LSD and he presses on the right lever, he'll get a little rat candy in the center of the center dispenser. If he presses placebo, if he gets a placebo, then he finds out that if he is at a placebo, that is just a saline injection, he presses the left lever and he gets a rat candy. So over 2 to 3 months, these rats reliably learn that if they're given, say, LSD, they press the right lever, they get a food pellet, press the left lever, nothing happens. And we could use different drugs. We've trained these rats with amphetamine, MDMA or ecstasy, LSD, all kinds of drugs. You can do anything that has a CNS effect in rats. This is a very powerful technique. And then just to prevent any positional preference, we train half the rats with the drug lever on the right and half the rats with a drug lever on the left. And so once we train the rats to do that, then we give them a new drug we've made that we think might be like LSD, might be a psychedelic. We give it to the rat and we observe, put him in the box and observe what does he do. And if he presses the lever he was trained on, he says, I think he gave me LSD. Now, that's actually a pretty weak readout on what these drugs actually do, because if you give LSD to a human, they don't just say, I think you gave me LSD. I mean, all kinds of other things happen. But this gave us a good number. We had typically 12 to 15 rats trained, and we would give increasing doses, and we'd find doses where half of the rats would think they'd been given LSD, for example, and that would be an ED50. And we could compare the ED50s of different drugs, and that would give us a measure of how potent it was in producing LSD-like effects in the rats. And so that was the main thing that we used for many years. Now, rats, when we first started using them, only cost us $2 or $3. They weren't particularly expensive. We had colonies of rats, so we typically had 5 or 6 colonies all the time. So it'd be anywhere from 50 to 90 rats in our colonies. Rats got more expensive over time, like everything in science did. Now rats are $50 or $60 a piece, and 2 to 3 months of training. That's a real pain. So the alternative is to use mice. So mice are now about $2 to $3. That's my understanding. They cost about what rats used to cost. And they do a funny thing called a mouse head twitch. If you give a mouse a psychedelic, he shakes his head back and forth, side to side. And so Adam Halberstadt, a scientist at UC San Diego, found out that if you implanted a powerful magnet under the scalp and cemented it to the skull, sutured it back up and let it heal, now put these mice into a big glass tube that was about 6 inches in diameter and wrapped some wraps of copper wire around to make a coil. You know what happens if you move a magnet inside of a copper coil? It generates a current. So initially, you'd have to look at these mice. They video record them. Some people still video record them if they if they do this assay, and some people do. And you'd have to have a graduate student or technician sit there and count every time the mouse would shake his head. That's not very efficient. And if you want to scale it up and do multiple drugs, it's really not efficient. So if you put a magnet under the scalp and put them in that tube, when they shake, what you get is it generate a current like this. They shake their head back and forth. Now that's been modified by a guy at Virginia Commonwealth, Javier Gonzalez Meso. He just hooked a magnet to the ear. The same thing happens. If he shakes his ear, that magnet swings inside the coil and you generate a current. And so using software, you can reduce this down to a single pulse. And just to show you, here is DOI with a dose response curve, placebo, and increasing doses. And what you can see is when you just record that head shake as a single spike, you can see you get increased numbers of spike. And of course, you can quantify those. And that'll give you a dose response curve, number of shakes over time in response to dose. And if you convert it into a curve, here's LSD with vehicle showing nothing here. And then as you increase the dose, and then you can integrate under the curve here. So this is a more typical way to do it now because mice are inexpensive and you don't have to train them. Now, why does activation of serotonin 2A receptors have any effect on consciousness and perception? If you actually look in the brain, I showed you that earlier slide, the PET image, 5-HT2A receptors are the most widely expressed throughout the brain. And they're expressed in key areas. So this is the frontal cortex. Here are those pyramidal cells. And the apical dendrites here highly express the serotonin 2A receptors. But they're also expressed in a lot of other areas. In the reticular nucleus is an area that wrapped around the thalamus. The thalamus actually is sort of a gate that decides what gets sent up to the cortex. And so activation of 2A receptors in the reticular nucleus, that has inhibitory projections into the thalamus, so you can regulate information that gets sent from the thalamus to the cortex. Also, the locus ceruleus is a brainstem nucleus that sends noradrenergic projections up to that same area. Norepi alpha-1 receptors are expressed in the same area that apical dendrites as the serotonin 2A receptors. And so they're modified also by activation of 2A receptors. The Raphe nucleus projects all the serotonin up to higher forebrain centers. And it's also regulated, its activity is regulated by serotonin 2A receptors. And the ventral tegmental area is a dopamine cell body that sends excitatory dopamine up here. So all these different areas, and somatosensory cortex, prefrontal cortex, and locus ceruleus, Raphe, and ventral tegmental area, their activity is affected by serotonin 2A receptors. So everything that's involved in cognition and perception, memory, imagination, etc., is affected by modulating the serotonin 2A receptors. Now, just to show you, this is a real-time using magnetoencephalography. This is what your brain normally looks like, and you can notice from the time signature here, 700, 800 milliseconds. So this is slowed down quite a bit so you can see what happens. But your brain is constantly sending these electrical signals, processing these signals through the brain, and that's the way things are transmitted. So this was done years ago by a group in Imperial College in London. So what they found is normally under placebo, those currents that I just showed you, you have specific anatomical areas that are connected to each other through long range and short range connections. You have a lot of intra connections and also interconnections. But basically, if you imagine that these are organs in the brain, and this doesn't correspond to where they are, this is just an illustration. You have areas in the brain that have communications within that area, but also reach out occasionally to other areas to send signals to other areas. So they influence other parts of the brain. This is what normally happens. But under the effects of psilocybin, the connectivity within those anatomical regions or hubs is broken down, and they reach out and start connecting with all these other areas. So you have this dramatic increase in global connectivity. So all these areas start talking to each other in ways that they normally wouldn't. So the visual cortex, for example, in the back of the brain, normally processes visual information. But under the effect of LSD, its connectivity expands out into other areas of the brain. So people would say that psychedelics cause mind expansion. This is almost literally true if you think about the way that this connectivity expands out to a kind of global connectivity. Then the drug wears off and it drops back to this normal placebo state where you don't have all this high level of interconnectivity. So in 2017, we solved the crystal structure of LSD in the serotonin 2B receptor. 3 years later, we solved it in the 2A receptor. But just to show you, for example, one of the things we had found is when I was still at Purdue and one of my graduate students looked at was this is the loop, extracellular loop 2, that hooks the top of helix 4 to the top of helix 5 in the receptor. And we noticed that there was a leucine in that loop, leucine 229, that looked like it could interact with the diethylamide part of LSD. And this is just a blown up view. Here's leucine 229. Here's the diethyl groups of LSD are docked in there through simulated docking with a computational routine. So this is the actual crystal structure shown in space-filling molecules. And this is extracellular loop 2 that goes from the top of helix 4 to the top of helix 5. And this is at leucine 229. You can see it's turned down. Most of these residues are turned out toward the extracellular space. Leucine 229 is folded down. Here's LSD. There's also a tryptophan here, which you can't see. And this leucine 229 wedges LSD into the receptor. So what we did was we wondered, how important is that? So if you look at the receptor kinetics, so this is what happens if you take a receptor preparation. These are 2A receptors expressed in HEK, human embryonic kidney cells. You put radioactive LSD in there, you let it equilibrate at 37 degrees Celsius, and it takes 2 to 3 hours to equilibrate. And then you remove the supernatant and take out the radioactive LSD, put in fresh buffer with no radioactive LSD, and you take aliquots out at various times and to look at how fast the LSD dissociates from the receptor. So this is the wild type receptor here, and this is this dissociation. So this is LSD coming back out of the receptor. And you see that it doesn't get back down to baseline to between 6 and 8 hours. So LSD lasts for 6 to 8 hours or longer. Also, it takes 2 to 3 hours to equilibrate because LSD has to get around that leucine, get into the binding site, and then once it gets in, it's kind of trapped in there. So we thought, okay, what happens if we mutate that leucine to an alanine? You know, an alanine is very small. It's the smallest amino acid except for glycine. And the leucine has a big, bulky hydrophobic piece out there. So we made that residue smaller and took that mutated receptor and did the same experiment. And look how fast the LSD came out. It's back out within about 4 hours as compared with the wild type. The other thing we did is look at canonical signaling. One of the things that the G protein signaling does is produce increased calcium. And for the wild type and the leucine alanine mutant, there's really no difference. But when we looked at beta arrestin recruitment, that other signaling molecule, you see the wild type arrestin is quite potent and efficacious, whereas in the mutant, where we make that smaller amino acid turn the leucine into an alanine, it doesn't have the same effect. And also, the arrestin recruitment is time dependent. So if you look at 5 minutes of incubation versus 5 hours of incubation, see the longer the LSD is in there, the more potent it becomes. So the dose response curve shifts to the left and the efficacy also increases. But if you look at calcium, you don't see any difference. Or if you turn that to an alanine, there's no difference. So this showed us that the recruitment of arrestin was related to that amino acid trapping LSD within the receptor. Now, I don't know if you want me to go off and talk about MDMA, I don't know how long you want this to run. That would be my psychedelic talk. I could talk about MDMA and ecstasy, depending on what your time constraints are, or I could just stop now and take questions. So
I think we have no time constraints. So if you want to talk about it, we would love to hear you talk about it.
okay, so I can talk about ecstasy, which I named as an entactogen. That's the name that I've been trying to promote as a class of psychopharmacological agent that's different from the psychedelics. And this is the structure of the S isomer of MDMA, methylene 3,4, methylene dioxy. This is a methylene dioxy. Again, an aromatic ring, 2 carbons away from a basic nitrogen with an alpha methyl. The alpha methyl prevents monoamine oxidase from degrading the side chain. So this is a card a friend of mine sent me years ago. Life is beautiful. The world is beautiful. I am beautiful. Have you been taking Prozac? Heck no, it's ecstasy for me. And by the way, you're beautiful too. This picture is about 40 or 50 years old. I made all the ecstasy that MAPS used for their phase 1 and phase 2 studies. We made 1.4 kilograms of high purity MDMA. This is actually me working up in the lab back when my hair wasn't so gray. My beard had a little bit of brown left in it. And this was one of about 3 flasks we had of this batch of ecstasy. So the DEA, when ecstasy first got really popular, people were taking it at raves. I'm sure it must still be out there. It's also called Molly. The DEA said it's just another hallucinogenic amphetamine. And of course, hallucinogenic amphetamines were Schedule I controlled substances. And I didn't think that was true because I had done a lot of work on psychedelics and also amphetamines. I actually had a patent on making the isomers of psychedelic amphetamines. So I thought, I'll prove that this is not a psychedelic, just another hallucinogenic amphetamine. So this is DOM or STP. And the more active isomer has the R configuration here at this asymmetric carbon. And I've shown it here, I've circled it with green. SDOM, if we reverse that, is inactive. And yet SMDA, with the stereochemistry like RDOM, so it's S versus R, it's active. And it's an entactogen. A lot of people don't know about SMDA. Racemic MDA is called a psychedelic or hallucinogen, but the S isomer is really a short-acting entactogen. So DOM or STP is a primary amine. It's just an NH2. If you add a methyl group here, it kills its activity. So this is R, the R isomer still, N-methyl DOM, and it's inactive. And yet, if we put a methyl on MDA, we have MDMA, and it's active. And it's the S configuration. So this is already making a case for the fact that the psychedelic amphetamine, DOM, is not like MDA or MDMA. But another trick we wanted to use was Sasha Shogun had made the alpha ethyl analog of DOM. He called it BL3912A. And he had proposed that it might be an antidepressant of some kind. And he was a consultant for Bristol Myers Squibb, and so he told him about it. So they made a big batch of this stuff and tested it for antidepressant activity. Now, whereas DOM is active in the 5 to 10 milligram range, if you extend this alpha methyl to an alpha ethyl, it loses its activity. And they inadvertently dosed a woman. They took this into an assay in geriatric senility. These were people that had cognitive deficits, geriatric people with cognitive deficits. They inadvertently gave this woman something like 240 milligrams of this alpha ethyl compound, BL3912A. Again, 5 to 10 milligrams of DOM is active, but if you extend the alpha methyl to an alpha ethyl, where you have a methyl ethyl, the compound is really dead. More than 100-fold less active. And yet, if you put the alpha methyl onto MDMA, it's an entactogen. And it's again the S isomer. So you see this similarity. So this convinced me that it wasn't just another hallucinogenic amphetamine. The stereochemistry was reversed. It had an N-methyl. And when we put an alpha ethyl on, it retained its activity as an entactogen, and there's no way it bound to psychedelic receptors and produced psychedelic effects. In fact, it doesn't. And so another thing we did, I had hooked up with a guy named Wilfried Dimfel in Germany, and he had been using EEG in humans to determine the onset of drugs. So he would have people come in and they would say, well, how long does it take for Valium to start having an effect? So he'd bring people into his laboratory, hook them up to an EEG cap, and he'd give them Valium, and then he would measure changes in their EEG, and so he could quantify it. And I said, so could we do qualitative tests? We could compare different compounds in the same way in rats? Sure. So we compared amphetamine, LSD, and MDMA in rats that had 4 different electrodes implanted in their brains, in the frontal cortex, hippocampus, striatum, and reticular formation. And these are different frequencies, delta, theta, alpha 1, alpha 2, beta 1, beta 2, different frequency domains. And so in these 4 electrodes and 6 domains, you really have essentially 6 times 4 variables for each one. So if you look at amphetamine versus LSD versus MDMA, you see some major differences. So again, looking at electrical activity in the brains of these rats, we could say, this doesn't look like LSD at all. I mean, if you look at, alpha 1 here, it decreases in MDMA, increases in LSD, decreases here in this hippocampus, but increases in rats here. It increases here in the striatum, but decreases here, and there's a big decrease here. So the electrical activity in the brain of these rats was very different for MDMA than it was for LSD or for the psychostimulant amphetamine. So what it turned out, and we did the first work on this in 1982, it turns out that what MDMA does, it's a false transmitter, and it's taken up in the serotonin reuptake transporter in neurons. So normally after a neuron fires, if this is a serotonin neuron, it has serotonin stored in vesicles, which is released into the synapse. It diffuses across and activates postsynaptic receptors. So amphetamine, if it was a dopamine neuron, amphetamine would would displace dopamine and norepinephrine and cause a release. So MDMA gets in through the serotonin transporter. It's a substrate, false transmitter. It's taken up and it displaces intraneuronal serotonin. And then the serotonin is released from these vesicles, and it's unrelated to the firing, the action potentials. It's a drug-induced release. So MDMA does that. It releases serotonin. It also releases, to a certain extent, dopamine and also norepinephrine. So MDMA is an indirect releasing agent that activates these postsynaptic receptors by the presynaptic release of serotonin, norepinephrine, and dopamine. So this is serotonin, release of tritiated serotonin. So we did this by looking at tissue slices. So we took rat hippocampal slices, incubated them with radioactive serotonin, put them into a superfusion bath that we had developed. And so then we wait till the efflux of tritiated serotonin has leveled off. And then we put in various drugs. So we had MDA, MDMA, and MBDB. Now, with respect to serotonin from superfused rat hippocampus slices, you see MDA is the most potent. And then we have MDMA and MBDB. And so both MDMA and MBDB are about equal potent in releasing serotonin. When we look at dopamine from rat caudate slices, dopamine is sort of the feel-good chemical. So people who take MDMA, they like the euphoria that it produces, which is related to dopamine and norepinephrine release. So again, looking at the percent released of dopamine, radioactive tritiated dopamine from rat caudate slices, you see MDA is a good releaser of dopamine. MDMA is much weaker, and MBDB does not release dopamine at all. We published on MBDB. It showed up in one time. They made a seizure of it someplace in Europe, but it never did show up anywhere else. There were never any other seizures because this dopamine release with MDA and MDMA is what makes you feel the euphoria of the drug. It's what makes people get up and feel stimulated and dance and all that. MBDB didn't do that. It never became a popular recreational drug because it didn't produce the kind of euphoria that you saw with MDMA or even MDA. And extracellular dopamine release in the nucleus accumbens correlates with reward properties. Again, this is amphetamine, methamphetamine. They're reinforcing people take methamphetamine, they want to keep taking it because it releases dopamine in the nucleus accumbens, which is the reward center of the brain. So here's MDMA. You can see it gives a good release of dopamine from nucleus accumbens. MBDB is down here, it doesn't do anything. MMAI is a compound we made that also had MDMA-like effects in rats, and fenfluramine, which was an appetite suppressant. So of all these 4 drugs, only MDMA releases dopamine, and it's the only one that produces any kind of pleasurable effect. MBDB doesn't, MMAI doesn't, and fenfluramine doesn't. We also looked at the ability of MDMA to release norepinephrine. A lot of people had not looked at that. And so these are plots from a chamber we had that we bought, and they have photo beams and photocells across here. And as the rat moves around at various times, the photo beam is broken by the rat, and it'll mark where the rat is when that photo beam is broken. So if you just put a normal rat in here, they wander around the cage and they break the photo beam. It looks kind of like this. If you give these rats 2 milligrams per kilogram of plus dextroamphetamine sulfate, the rats just run around like hell. You can see they're breaking the beam all the time. It's a psychomotor stimulant. If you give MDMA, and you see the dose is lower, we give 2 milligrams of dextroamphetamine here, 5 milligrams of racemic MDMA, but you see it still stimulates activity in the rats. But then we injected the rats with prazosin, which is a noradrenergic alpha-1 receptor blocker. And when we give them prazosin, it shuts it back down. And prazosin is selective for norepinephrine. So this was an indication that MDMA also released norepinephrine to produce its CNS stimulant effects. So just mentioned briefly, I started the Hefter Research Institute in 1993. We're the ones that supported most of the research that people have seen that got them interested in psychedelics. We supported Charlie Grobe's study of MDMA or of psilocybin in terminal end-of-life cancer patients. We funded the studies at New York University, Johns Hopkins University of psilocybin, again in cancer patients. Funded a study of Michael Boganchutz looking at psilocybin treatment of alcohol use disorder, alcoholism, and showed that was effective. So then the government didn't fund any of these. In fact, the government people still think that these drugs are dangerous drugs that should never be used. And that attitude is slowly changing because what they've seen is these clinical trials where people that have treatment-resistant depression, nothing else works. Psilocybin has worked, and in people with PTSD, MDMA works. So we actually I started this 1993 when we funded $10 million total support of mostly seed money to get these projects started because the government wouldn't fund them. So just to acknowledge, I didn't do all this work. I had lots of graduate students. I always had lots of students who wanted to work for me. For some reason, they were interested in studying the chemistry and pharmacology of psychedelics. And in fact, one of the faculty members complained once about, they said, why does Nichols get all the graduate students? So I thought that was kind of a tribute. They liked working for me. So just to acknowledge a lot of work. And also I had a grant from National Institute of Drug Abuse for 28 years, and I got a 1-year extension to study specifically the chemistry and pharmacology of psychedelics. In my last several years at Purdue, I was a recipient of Robert C. and Sharpie Anderson Endowment for my work as a pharmacologist. So with that, be happy to take some questions if you have any.
Wow. Thank you so much for that.
I hope that's what you were hoping I would talk about or something like that.
Sorry, what did you say?
I hope that's what you were looking for.
Oh yeah, it was everything we were looking for. And then you gave us some more. It's like
Right.
so grateful. I think we do have a few questions here in the room. We're gonna start off with Jacob over here. By the way, my name's Rev. I forgot to introduce myself. I'm sorry about that.
It says Jacob though on your
Yeah, so Jacob is like this guy that's coming up right now.
Hi. Yes, sorry about that. I'm dealing with like a medical thing. But I was gonna ask, could you just briefly talk about cannabis and psychosis? Like anything you've seen about cannabis and psychosis?
I haven't done any work with cannabis. Of course, it hits cannabinoid 1 receptors. That's its mechanism of action. I know that people who use it will sometimes develop psychosis. That's probably a pre-existing condition. I don't know much beyond that. I don't know what the incidence is. I can tell you that the illegalization of cannabis has made the recreational use of cannabis more dangerous than it ever was. Because when I was a graduate student at the University of Iowa, the high potency cannabis that you could get was something like Colombian yellow, which had like 2 or 3% THC, which was supposed to be really potent. And now I was looking at some strains the other day. There was one called watermelon that they claimed had up to 24% THC. So I think with these higher potency strains, you have people just using them willy-nilly. And of course, it's safer than alcohol, but still high potency cannabis can produce panic anxiety reactions. A lot of people have panic anxiety reactions. And it doesn't surprise me, I guess, that some people, especially with high potency cannabis, can develop psychosis. The other thing that's kind of insidious about cannabis is, whereas people typically won't drink alcohol all day, they'll maybe come home and have a beer or two, or watch a game and have a beer, maybe on weekends go to a party and have a few beers. People that use a lot of cannabis typically smoke a lot of it. They'll smoke it in the morning, smoke it several times during the day, smoke it at night. So it's really a chronic thing for a lot of people. And I think that's probably also contributes to the development of cannabis psychosis. I don't think that any of these drugs actually produce mental illness per se, but if you constantly change your consciousness, you constantly are changing your perceptions, and you have a predisposition, you probably can develop it. The same thing is true with psychedelics, with LSD. You have people that are predisposed to psychosis. The incidence of schizophrenia, they diagnose it typically late adolescence, early adulthood. And so it would be a typical thing that somebody might send their student off to college, and they've never been away from home before. They're in a strange situation. They don't really know what the routine is. And they develop schizophrenia because of the stress. The stress can precipitate it. And it can also be precipitated by people using psychoactive drugs like psychedelics. So you have to have a predisposition. I would assume that's probably true for cannabis. It's just that a lot more people are using high doses and large amounts of cannabis. So it's probably something they see more often. And the other thing is there's a general perception that cannabis is pretty safe. And certainly it's one of the safest psychoactive drugs known. I mean, nobody's died of an overdose as far as I know of cannabis. But this perception that it's safe leads people, I think, to use it excessively. And I think anything done to excess is not good. But I can't give you chapter and verse as far as why cannabis would cause psychosis. It's probably there are people that are labile or not stable to begin with. And a lot of times those people, I went to a conference once on psychedelics, and there were people there that had pink hair and multiple body piercings and tattoos all over them and whatever, and expressing their individuality. And they were all stepping out at breaks to smoke cannabis. And I told a colleague of mine, I said, these kind of people probably shouldn't be using psychoactive drugs. They don't look like they're put together right. So there's nobody that's covered with tattoos there. But I wondered about some of the people that were using these drugs.
Thank you for that. We have another question over here. He was first.
Can you hear me?
I think it's better if you come up. You should introduce yourself too.
Hi, my name is Ben Reinsma. I saw the Ultra LSD episode on Hamilton's Pharmacopoeia, and I was just wondering some of the newer drugs that you have developed since then, since you've got the 3D model and with a computer program. showing the 5-HT2A receptor.
Yeah, I haven't done much lab work myself. When the pandemic started, nobody was going into the lab, everybody's working from home. So I've had access. I dial into Zoom group meetings. I had a heart attack in February 2019, which compounded situation. And then I fell down last year and hit my head in the concrete floor of my garage and gave myself a subdural hematoma, which was successfully drained, and I'm back to decent health. But I haven't been in the lab working much. And I worked for a company as a consultant known as Elucis Therapeutics. That was recently acquired by Beckley Psytech, and now I'm working for a new company. And I've used some of the information about these receptors to design some new molecules. We designed one. If you look at those models, if you look at the model for in the 2A receptor, we published it for a 25 CN ENBO. And when you look at the structure, the 5-methoxy is just sticking out into nowhere because the resolution on the structure is not high enough. And I proposed that it was interacting with an asparagine at the top of helix 6, asparagine, I think 343. And I designed a molecule with a tether that would stretch over to make up for that water molecule. So you didn't need a structural water there for it to bind. And that molecule we've done preliminary testing on and shown that it's actually active. So, and the lab where I'm working now, Brian Roth got a big grant from DARPA, Defense Advanced Research Projects Initiative, whatever. And they've used that to, and just published a paper recently on the results of their early results from that, where they virtually did docking studies by Brian Schweikett at UC, I think San Francisco, and discovered some new chemotypes that people had not been aware of before that bound to and activated that receptor. So having access to the receptor itself, if you had the right library of compounds, you could dock it to that receptor and get docking scores and find new molecules. They published one recently. I forget the journal. I'd have to go back and look. But they published it. One of the early molecules, the first molecule, proposed that it had antidepressant effects, but in a mouse model did not have behavioral effects. So there is work to use those receptors, to publish receptors, to dock things and develop new molecules. I can't talk about what they've developed because they haven't filed a patent on it yet. But there are compounds that have been developed with that technology. And then I used the published structure to develop some molecules that we're developing as anti-inflammatory agents that have to engage that asparagine residue at the top of helix 6.
Very cool. Thank you.
Sure.
Cool. We don't want to hold you up any like a lot or anything though. Do you still have time to take a few more questions?
Okay, what time is it? 7:30? I could take a couple more. If there
Okay. We have one over here. Yeah, Ricky.
Hello, my name is Ricky.
What's your name?
Ricky Richens. Nice to meet you. I'm really curious why it is with lysergamides, when you take they're a substituent group that the ambient nitrogen is attached to, why the difference in that can result in gangrene or something as euphoric and psychedelic as LSD. Like, do we know why changing a simple constituent group can do so much on the same compound?
So we know that the ergot alkaloids actually bind pretty tightly and stay on the receptor for a long time. For the ones that are vasoconstrictors, basically they're activating alpha receptors that are in the vasculature. When you, like ergotamine, And when you change that substituent, you change the receptors that it binds to. So LSD loses its affinity for those alpha receptors that cause vasoconstriction, and it just activates the 5-HT2A serotonin receptor. And you don't have serotonin receptors. The 5-HT2A receptors are not critical for maintaining blood pressure, tone, blood pressure, heart rate, or anything like that. So it's mainly effective. By changing that substituent, you've changed the receptor variety of receptors that it can bind to. There are other lysergamides that are used clinically to prevent postpartum hemorrhage, for example. After childbirth, and when the placenta comes out, there's a lot of blood tearing of blood vessels. And in earlier eras, the biggest loss of life in childbirth was the mother bleeding and then having severe blood loss and bleeding. And so you have to stop that. So even today, they'll use ergot derivatives, specific derivatives that cause vasoconstriction, but in low doses. But it's the difference between LSD and something like ergotamine or one of the others, or ergonovine. The difference is that substituent changes the nature of the receptor. So ergot alkaloids bind to a lot of receptors. LSD, you showed that slide, it binds to dopamine D1 and D2 receptors and alpha receptors, et cetera. But its main activity is binding to serotonin 2A receptor. So when you change that substituent, you can modulate the receptors that ergot then binds to. All the ergot derivatives usually have high affinity, bind tightly to their receptors. So if you have something that activates a receptor like adrenergic receptors that cause vasoconstriction, constriction of blood vessels, if it sticks in there for a long time, or you keep if you kept taking it chronically, then you would get gangrene because it would block, the blood vessels would stay constricted. LSD doesn't have that particular type of activity.
Thank you very much.
Answer your question?
Yeah, definitely.
Okay, great.
But I want to let people talk. Thank you, though.
Nick? Yeah, sorry, other Nick.
Hello, my name's Nick, and so in the majority of clinical trials that I've seen, they're employing like methylphenidate or dextromethorphan, and I'm noticing that these papers are either not reporting the success of their masking or reporting a really low success rate of the masking. So I guess I'm wondering, like, what is an ideal placebo for these types of substances?
Nice. If you know what it is, I'll be happy to pass that information. The problem is, this is a chronic problem that's going on for many years. What is an effective placebo? If you look at a standard SSRI, like Prozac or something like that, it has no effects that can be detected by the person. So the standard for FDA clinical trials is a randomized placebo-controlled clinical trial. So you randomize people to either placebo or the drug you're testing, and placebo or the drug. And if the most drugs that they test, I mean, you can imagine something like a benzodiazepine like Valium would have some effect, but it's not dramatic. It makes you feel less anxious. You feel kind of relaxed, etc. You might be able to pick up the difference between a placebo and something like Valium. But generally, in most of the placebo-controlled trials, the objective is to do everything else, set up everything else the same protocol, the same treatment facilities, the same, for these clinical trials, they have a whole protocol written up. This is the way you have to do it. At a certain time you do this, a certain time you do this, etc. Well, with a psychedelic, the effects are so profound in general that the person who has taken the actual psychedelic knows that they got the psychedelic, and the person who got the placebo knows they didn't get the psychedelic. I think there was a study that was done at Johns Hopkins, maybe it was the 2006 study, where they used methylphenidate as the quote unquote placebo, and they used a high dose of it. In Michael Bogenschutz's recent study of psilocybin treating alcohol use disorder, he used a big dose of Benadryl, which is an antihistamine that produces sedation. So if you have people who don't know what a psychedelic is, you might be able to fake a few of them, like the high dose methylphenidate studies. They had people who were the sitters or the therapist. They would ask them, the person you sat with today, did they get placebo or did they get the psilocybin? And the same thing is true with the Benadryl in the studies Michael Bogenschutz did. They'll say, well, did you get psilocybin or did you get the placebo? And at least in the methylphenidate case, there were a certain percentage of patients that thought they got the actual drug. And there were even some of the guides that thought that the methylphenidate subjects got the drug. But then they broke the blind. It turned out they didn't. So it was a low ability to discriminate in high doses of methylphenidate or Benadryl. But basically, this is a continuing problem. You really want to have a placebo that is just like the drug you're trying to test, like an SSRI. Well, you give somebody a capsule full of lactose, wouldn't do anything, or a capsule of Prozac, they wouldn't know the difference in general. Because SSRIs would take 2 to 3 weeks to really work. So you'd have a big clinical trial, you'd enroll 300 people, and you'd give half of them a placebo and half of them Prozac. And then at the end of that time, you'd see who had their depression alleviated. But otherwise, there was no basis for discrimination of what they were. But with psychedelics, give somebody, 20 milligrams of psilocybin, and if they have any experience with psychedelics, they know exactly what they've gotten. Most of the clinical trials that they've done, they've tried to find people that have no experience with psychedelics, which would assist to a certain extent in blinding. But it's a question that gets asked over and over. It's a question nobody can answer. What's a good placebo for a psychedelic clinical trial? And as far as I know, there isn't one.
Okay, cool. Thank you.
Yep.
So I think we have how much more time do you have, Dr. Nichols? We have like 3 more people trying to ask questions. Do you have time for that? Maybe like 1 or 2 more?
Yeah, let's do 2 more.
Okay. Nick, other Nick.
Hello, Dr. Nichols. My name is Nicholas Denton. I want to thank you for coming in here. You're definitely somebody that I look up to. I first read TCAL and read the introduction, and I thought...
I wrote the introduction to PCAL, yeah.
TCAL, sorry, that's what I meant to say. And I just think it's really refreshing to look at psychedelics as a medicinal tool professionally like this. And in that I kind of think about what is the correct medicinal application of psychedelic medicines. And I look like to the traditional cultures a lot as well and see what they're doing. And they talk a lot about, like you said, in the definition of a psychedelic, there is the difference between the transcendental or the religious experience and then the psychedelic experience and the mystical experience. And they're all kind of tied together. One thing that was in there as well was dreaming. And one thing I've seen in the studies is talks about 1-irigens, which are plants that are classified to enhance dreaming activity. And I realize there's a lot of psychedelics that kind of overlay into there, and that your definition also included dreaming in psychedelics. So I was just wondering really if you could speak on the pharmacology or if there is any medicinal application or any kind of clinical research into compounds that affect the dreaming consciousness, or if there's nothing there, because I read the literature, there's not a lot. But if there's nothing there, then also like the differentiation between the mystical experience and the religious experience versus the psychedelic experience, and why they don't always go hand in hand.
There was a paper that was published years ago by a woman named Clara Torta. And she took, I think, 2 volunteers into a sleep lab. And then when they were asleep, slowly infused the low doses of LSD. And in both cases, the EEG went into REM sleep. So it induced dreaming right away. That's the only time I've ever seen anything like that. I think when you want to talk about differentiating a religious experience from a psychedelic experience, it's really not possible, because the people that, I talked to a student here at UNC a couple years ago. He had graduated, he had a degree in, I think, electrical engineering. He had been graduated summa cum laude or whatever, and he didn't know what he wanted to do, but he'd heard about psychedelics. So he came to my office, and what he told me was, I read a lot of literature about psychedelics, and I want to have a psychedelic experience. And I went to this music festival, and I took a substance. He didn't tell me what it was. I suspect it was LSD. He said, I took a substance. I thought at the music festival, there would be lots of people who would be able to take care of me if I had a problem. And I was sitting up on the grass above this band playing. And he says, when the substance kicked in, I looked down at the grass, and the grass turned into little people running all around. And then I knew, I sank into the ground, and then I was projected up in the sky, and I was looking down at my body in this place. And he said there was this intense feeling of love and of being, sort of part of everything. And so he decided he wanted to use these to treat people. He says, I see that they're using these now to study their medical application. He said, so what do I need to do? Because that's what I want to do with my life now. I want to treat people with psychedelics. And his mother was divorced, and he told her what he did. He shared everything with her, and she was shocked at first. And he said, no, Mom, this is the greatest thing that ever happened to me. It showed me what I want to do. I worked with another guy who became a psychiatrist. And that's why I told this guy, I said, well, in a few years, these things may be legal. Go to medical school, get a degree in psychiatry, find somebody who's using them, and then there may be a place for you. But then I worked with another guy years ago that was a psychiatrist, and he was from Texas, actually. And he said he took a psychedelic, and he was trying to figure out what he wanted to do. He didn't know, he was nearing his senior year in college. And this big neon light lit up in his eyes. It said, become a psychiatrist. So he went to medical school and became a very successful psychiatrist. So I'm not sure, there's an overlap there. For people who have taken, say, 5-methoxy-DMT, which some people have called the God molecule, that experience will put people in touch with some kind of a being, a godlike being, and people who have been atheists sometimes say, I'm not sure I'm an atheist anymore because of that experience. So it gets kind of very difficult to discriminate when you say a psychedelic experience versus a religious experience. In treating people at end of life, the first study that Charlie Grove did, which we had to take down because the woman's husband never got over her death, but she had a melanoma in her intestines. I didn't even know you could get them in your intestines. She had a melanoma, and they didn't completely resect it. And he got the slides and when you take a tumor out of something like that, you have to resect out past the margin of the tumor. If you leave any bit of the tumor there, it'll just grow back. And they hadn't resected out past the margin. She was in California, and the medical center didn't dissect it out past the margin. And he got really upset. He went back, says, you need to go back in, and they refused to do it. So he sued him, and after she died, won, I think, $2 or $3 million lawsuit. But anyway, so we had a video of her, and she became very depressed when she got the cancer diagnosis. She quit doing all the things. She was an executive with the Educational Testing Service, went around the world setting up, the GRE tests and things like that. It was an Asian woman. And so when she came out, I asked her husband, I said, could you see a difference in her? He said, she came walking down the hall and she was just beaming with this big smile. The difference is like night and day when she went in, when she came back out. So she had an experience where she said she had seen her cancer as this big formless black thing. And she looked at it and she said, I'm not ready for you yet. I still have life to live. And it kind of disappeared. And that all of a sudden she felt good again. She said, I'm not dead yet. I still have life to live. Why should I be depressed? Completely changed her life. Now, it was a life-changing experience. People that I'm really an advocate for people treating dying people who are, death is really an existential distress that many people just don't deal well with. And for people who've been treated with cancer with the psychedelic, psilocybin especially, they lose their fear of death. They want to get in contact with estranged siblings and so forth. Their whole attitude toward life completely changes. So, was that a religious experience that transformed their lives, or what exactly was it? So I'm not sure how you can differentiate, what is a religious experience or mystical experience versus a psychedelic experience, because a high dose psychedelic experience, almost by definition, is a mystical experience. I'm not a philosopher, so that's probably the best I can do.
That was a great answer. Thank you very much for taking your time.
Sure.
All right, Dr. Nichols, I have the final concluding question. It's kind of like a two-part question. It's half informal, half formal. The informal part, have you, Dr. Nichols, personally have had your own profound psychedelic experience? And the second question, for students here, mostly me, interested in neurochemistry of psychedelics research, how do you advise on we can get involved in that kind of stuff?
So when I was a professor at Purdue, I never talked about my personal experiments.
Yes.
I was in a pharmacy school, but and I don't have anywhere near the experience of someone like Sasha Shulgin or probably most people on the street. Over the years, I was very cautious. I did a few things, and some of the experiences, one of the experiences in particular that I had, convinced me that these drugs really were very useful therapeutic agents. In my life, I probably haven't had more than 10 or 12 experiences with different things, mostly with things that we'd made that I wondered if they were active. They weren't illegal because they were new compounds. So I was pretty cautious about that. But one experience I had, an early experience with LSD, where I visualized the universe. I was projected outside the known universe into what I can only describe as nothingness. If you think about where is the universe, it's in the middle of non-universe, right? And non-universe is infinite nothingness. And I know that sounds weird. And now I can't even visualize infinite nothingness, but I was projected outside the known universe. The universe was just like this light green glowing ball off in the distance in the middle of infinite nothingness. And I can say those words, but they don't have the same meaning. What does it mean to be in the middle of infinite nothingness? And at that moment, I had the perception that the function of the universe was to serve as a womb for life. And that obviously stuck with me. I still remember, like, the universe is full of life, obviously. And the experience I had, my father was kind of a very authoritarian, controlling person. And when I had that experience, I said, God, I wish my father could have this experience. I knew he never would, but I thought it would change his life. So I've had a couple of experiences, and I had one experience where I thought about why he was the way he was. And his father was a strict Pentecostal religious guy who beat all the kids and made them drop out of school and work on the farm when they were 13 or 14. He was the only one that was allowed to finish high school, and he went to the army. But I never knew, like, what was wrong with him. And I had an experience where I realized that he had never learned how to share. He never could share. And tears streamed down my face. I thought, oh my God, he never learned how to share. Now that sounds kind of silly, but at the time, it was really momentous for me because I was grappling with my relationship with him, why he was the way he was. And he had a very overbearing, mean, authoritarian father who literally beat the kids if they didn't behave. So, and he never shared. His mother never had a nickel to spend on herself. If you saw pictures of her, you'd look at her and you'd go, oh my God, look at that woman. She looks so sad. And, he was abusive to his wife. He was abusive to his kids. And I don't know what his father was like. He never talked about his own father. But I realized he was broken himself, had never learned how to share. So insights like that just made me realize, I saw another example of a student who was given MDMA. And I was in the group. Somebody went to Los Angeles and got a bunch of MDMA tablets and brought them back. And I was at this party and went into different rooms. And there was one room where they had some students in there. They were Purdue students. And the one woman was sitting in the middle and she started sobbing and crying. And everybody gathered around. It's like half a dozen people gathered around and said, like, what's wrong? What's wrong? And she says, I need to tell somebody something that happened to me and I've never been able to talk about it. And I said, and I was watching and they said, well, what is it? What is it? You can trust us. And they all were there and they had their arms on her, supporting her. She says, when I was in junior high school, I got pregnant and my parents didn't believe in abortion. They sent me away to live a couple states away to live with my grandmother till the baby was born, and then gave up the baby for adoption, and then came back and finished in my school. And she says, I've never told anybody that. I always wondered, did I do the right thing? What happened to my child? And I think she may have said it was even a daughter, I'm not sure, but she says, I really wondered what happened to her and if I'd done the right thing. And everybody around her was very empathic. This was the thing that really amazed me. They were so empathic. And he said, you absolutely did the right thing. You were like in junior high. You had no ability to raise a child. It would have ruined your life. It would have ruined her life. And now she's with somebody who wanted her, who could take care of her. And you're getting on with your life. And it was amazing the empathy and rapport that produced. So that wasn't something that I did, but something that I witnessed as, wow, this is really something, because you could get people to talk and open up about stuff that they'd never had. She'd never talked about. I mean, she was probably 22 years old and this happened to her when, junior high, I don't know, 13 years old, 14, I don't know. And she'd never been able to talk about it. And it had hurt her so much, the way her parents had treated her and having to give up the baby and all. So that was an example where I saw the therapeutic power of MDMA. And again, of course, it was used, quote unquote, recreationally, but really it wasn't recreational. And it just made, you could tell she felt so good having all these people that were her friends tell her, you did the right thing. You did what was best, and going ahead and telling her, like why you couldn't do it. She had got given up to parents that wanted her and . So I've seen things like that. And, I just become convinced that these things really, if they're used properly, can be very powerful. And, in ancient cultures, they use these things. The shaman would use them and to treat people with all kinds of things, all kinds of conditions. So we just lost the technology. And, nowadays you talk about having people eat magic mushrooms or taking ayahuasca or whatever. You go like, wow, yeah, we can give them Prozac, or we can give them Valium or whatever. And we've gotten away from that. But, this is ancient wisdom that we've kind of gotten disconnected from, I think. It just needs to be done the right way. And everybody's hoping that they don't screw it up again like they did in the 60s. So those are some skeletons from my closet, I guess. So I hope that was a productive evening for everybody. Maris, are you the faculty liaison or something, or?
Oh, no, I'm actually not the faculty liaison for this club. I'm just a faculty member in the program. And a lot of the students here in my class. That's weird.
Somebody's got an echo. Okay. So, thanks for the opportunity to proselytize, present some science, some personal information, and hopefully now you know more about psychedelics and entactogens than you did a couple hours ago.
It was really great to have you.
Yeah, thanks. All right. Okay, so
Have a good night.
all right.